Okay, hello everyone. Many thanks for joining this webinar provided by the RootCamp and KWS. And I'm very happy to introduce the participants. So let me share my screen. And first of all, many sorry that we are that late, but obviously we had technical issues, but everybody is on board right now. So let me share my screen. I hope everybody can see my screen now. The RootCamp provides a very short webinar series this summer just to show which topics are interesting for us, to provide information from our corporate partners. So I'm happy that today Chris Fender is joining from KWS. And so he's straight from the USA, providing their scouting for KWS. I'm very happy that we have another participant from the US. It's Alberto Acedo Becares, co-founder of Biome makers, microbiome company. Then more from this region. So we are located in Hanover, Germany, is Martin Kollmar. Part of his team is actually working from Germany. He is from Göttingen. It's in our way, and he will provide a new annotation method. And that is the youngest company here, not over close to a month old, I guess. And I'm also very happy to have Martin Spitznagel here, CEO and founder of BioX, and he will bring us his new product of biological weed control. Let me tell you just a quick intro on RootCamp. Who are we? What are we doing? So we are a multi-corporate incubator. We are now in the application period of our second acceleration batch, and our aim is to disrupt the agri-food value chain by connecting startups and corporate players. So we not only want to introduce each other, but really want to test the product and the technologies of the startups in industrial processes. As I said, we are located in Hanover, Germany. Our partners are our mother company, the accelerator Spin Lab from Leipzig, but also K+S had a great part in initiating us as an accelerator. We are very happy to also have KWS as a corporate partner, and this platform is provided by DLG, which is the organizer of the Agritechnica, the world's biggest agriculture fair. More partners are in our pipeline, and we will announce them in a very short time. So what are we actually doing? We offer an acceleration program of 3 months. We give a grant of 10,000 euros for the startups that are taking part of it. We offer a classical acceleration phase with many workshops, lots of coaching, but not only the educational part is our focus. We also have a side track where together with the corporate partners, the startups are preparing a concept on how they can actually use their synergies, and that is also being presented to a demo, and the qualifying startups are then invited to share also the second phase of our program, which is actually a pilot phase, which we also have a budget from up to 40,000 euros to actually test the technology, test the products the startups contribute within the industrial environment of the corporate, just as the intermediate phase. And the aim is surely to then establish a real corporate startup interaction, so commercial cooperation. And we see ourselves as a catalyst to actually support this process. What is our focus for the actual batch? So in general, we are looking for all verticals of the agri-food value chain. This time we are especially focusing on everything that's in biotechnology. We look for carbon farming technologies. We look for digital solutions and also for robotic solutions, and try to find the best startups in the early stage, everything it's pre-seed or seed, without regional limitation. What are we having? What is our plan? So right now is the application period till 17th of August. Please make sure that you apply on the platform F6S. We then have a structured selection process, and we will then start the acceleration phase 12 intense weeks from beginning of November, and the integration projects will start in February. Also in February, we will have the Agritechnica here in Hanover, and we are looking forward to then have a couple of satellite events and really have an able presence here during this huge fair. Also, one thing I don't want to miss out, next week, also on Thursday, we will have another webinar on carbon farming, where we also invite the corporate partner to share his view on challenges, and then have showcases of startups that are already looking into solutions and providing solutions to tackle these challenges. Now guess I need to change that. Now I'm happy to hand over to Chris Fender. As I said, Startup Scout for KWS in the US. Chris, please take over.
Thank you, Philip, and it's a pleasure to be here with you, and I'm very appreciative of the opportunity by RootCamp to facilitate this. I will share my screen, and I have a short presentation. There, can everyone, hopefully everyone can see the slides, and look forward to discussing a little bit about KWS, some of our needs, and start with a brief introduction of KWS for those that might not be quite as familiar with where we are. As a company, we are a global company. We have activities in over 70 countries around the world. We have over 5,700 employees, and we invest a significant percentage of our revenues into R&D, into creating new solutions for farmers. You know, you can see where we sort of rank with other players in the seed industry. And on this map, as you can see, we have breeding stations and field trials in many areas around the world. So it's important for us to look for technologies that allow us to be competitive on that scale. And so scalability is a key aspect when we're talking about a diverse range of markets and a diverse locations and regions in which we are active. Our portfolio of crops, you know, includes most of the major crops. The two that we're not active in are rice and sugarcane. Actually, sugar beet is a large crop for us, and really goes back to the origins of KWS in sugar beet. But we have expanded and are currently expanding even recently into vegetables. And additionally, other crops that are emerging that aren't on this list that we are active in, like peas, for example, in Europe especially, and opportunities that are emerging around plant-based proteins and utilizing peas. But for sure, we have a broad portfolio of crops that we are active in, and it's important for us to find technologies that, again, allow us to impact across crops as we want to be efficient with our efforts within R&D. A little bit more specific about the size of our R&D organization. Our main research campus is at our headquarters in Einbeck, which is not far from Hanover, as most of you know. I am actually located and work out of our St. Louis, Missouri, and in the Midwest, in the center of the U.S., the Gateway Research Center, as we call it there. It's located on the campus of the Donald Danforth Plant Science Research Center. We have around 40 researchers working there and working in a lot of the more basic research topics, but also still impacting in product development across a wide range of crops, looking at developing new tools that allow us to create new genetic variation for those crops. Again, we have a significant R&D presence, over 2,000 employees in R&D, and a considerable expenditure annually in supporting our R&D efforts. And that includes not only lab type R&D or what people are traditionally thinking of R&D, but obviously with the number of breeding locations we have, that's also a large part of our R&D efforts in developing new varieties for farmers. So we consider ourselves primarily a plant breeding company, and as opposed to companies that also provide other solutions or products for farmers. We were founded really in the plant breeding space, and are very proud to be a plant breeding company. So when we look for technologies, things that help us impact the plant breeding process are important to us. And my background, even personally, is more on the traditional plant breeding side as opposed to the more biotech side. So looking for technologies that help us be more efficient in creating genetic variation and selecting individuals within that variation that have the potential to be new varieties are important to us. And so there are a number of tools that we utilize in that toolbox to help us facilitate that process. And I'll walk through some of those here shortly. But with breeding in mind, and looking at the challenges that we're facing around the world, it's important for us to hold these as pillars in how we select varieties, to making sure that we're providing products for our customers and for farmers that give them the best chance to be profitable, create impact, and so looking for traits that provide resistance to disease and pests, and provide yield stability, and provide efficiency in using things like fertilizer and benefiting the soil, and then also with a changing climate, increasing stress tolerance for abiotic stresses such as drought or flood tolerance, even as the case may be. So some of the tools that we're using in order to help facilitate this is use of hybrid breeding and creating haploid lines and doubling haploid lines to help facilitate hybrid breeding. So we look for technologies in the tissue culture space that allow us to more efficiently generate doubled haploid lines that we can introduce into a hybrid breeding, and really take advantage of heterosis and the benefits that offers for many species, many crop species, of enhancing yield or enhancing other traits of importance that we just talked about. So technologies that help us with tissue culture, and those can even be broader than just doubled haploids, as well as we look at things like new breeding technologies and new breeding methods as well. So hybrid breeding schemes, creating instant inbred, so to speak, through the use of doubled haploids is important, and ways to help us facilitate that in the most efficient manner are of interest to us. Additionally, we employ a lot of marker-assisted selection to help accelerate our selections and really better characterize the genetic variation that we're creating, be more efficient with our selection processes, and really analyzing breadth of diversity that we're creating within our breeding programs. So, marker technologies are something that we employ. And again, these technologies that we would look for have to help us continue to scale our use of these tools to be more efficient than we currently are. Another area is in the digital space, and this is an interesting area for us as we look at the impact of environmental variation and really the impact that has on phenotype as we make selections in an increasingly diverse set of environmental conditions. So employing technologies that allow us to see things that we wouldn't normally be able to see within our fields, allow us to characterize things below the surface of what's happening in the environment for each plot, for example, and do that at scale. When you realize that we have the breeding activities around the world that we do, these technologies have to be able to be scaled. They generate a tremendous amount of data, and so there has to be the ability to analyze that data and make sense of that. So the analytics side is also of critical importance to us. But we do look for technologies in this space and how we can leverage the things that we've already developed more efficiently and at scale. And then new breeding methodology, creating new genetic diversity that would be difficult to create through traditional hybridizations or crosses between individuals, looking at ways to generate mutants through tilling, for example, or looking at ways to use other methodology to create a genetic diversity, or to better characterize the impact of existing diversity, the impact of a gene that we've noticed within our germplasm resources. How can we better characterize that using some of these new breeding methodology, and then integrating those more efficiently into our breeding schemes to get products to our customers faster than what we would have been able to do. And some of these I'm sure you're familiar with. There's certainly a increasing complexity of the regulatory landscape around these around the world, but using methods like CRISPR and other genome editing methods to create certain variants, and some of these are identical to nature induced sorts of variation. Others are not, but they again provide tools in our toolbox that we can utilize to hopefully better meet some of the needs that our customers and that agriculture has. And so you can see, when you introduce these new methodologies and these new tools that allow us to increase the amount of genetic variation that we have to work with, we can utilize them in different ways. We can develop varieties that have new traits, novel traits that haven't existed in nature, or we can use them to better characterize existing variation and select new varieties through traditional breeding schemes. So we are kind of we're certainly fully aware of the regulatory landscapes and respectful of that around the world. In the U.S., we have a little bit different regulatory landscape than what exists in Europe. So we do some of these new methodologies in the U.S., but nevertheless, they are of great interest to see what technologies exist that might help us facilitate that. And ultimately, with the goal of being able to, like I said, provide new solutions for farmers, for agriculture, that help overcome some of the hurdles that we exist. So it's one of the things that a partner like KWS, I think, brings to the table in terms of we have a lot of data around the markets, with the ability to introduce new product to a wide range of markets. We're seeing global challenges around the world as they begin to emerge. And we want to, in terms of agriculture being able to provide these things, I think it's a great time to be looking at new business models, new disruptive technologies in agriculture. And, as we talked about, a lot of these spaces below the selected fields that we have application with potential, precision farming, biologicals, robotics, the novel breeding technologies we just talked about, even synthetic biology and the opportunities that could possibly provide. And then ultimately, traceability, things that impact the consumer, that make the consumer feel more confident in their food supply are all of great importance to us. So when we look at how we would collaborate, with a startup, it is important for us to create a scenario that not only provides a win-win for us and the startup, but really a win for the farmer. We want the farmer to be successful. We work hard to maintain our independence, to allow the farmer to have choice, and so it is critical that we provide a pathway that the farmer and our customers can ultimately continue to be successful and be more successful than they currently are. We want to find ways to create added value and what we can bring to the table versus what a startup can bring to the table. And the agility that a startup brings to the table is very exciting. We were able to bring certain established technologies that we can deploy in conjunction with what a startup could bring to the table. And then this issue of scalability, I think, is incredibly critical when looking at, we have to be able to utilize new technologies at scale quickly in order to be efficient in providing solutions with our farmers. Startups are great because they are agile. They do break conventions and barriers. They think differently, and they really understand creating new technology. And so I think there's a lot of complementarities that we can work with when it comes to working with new startups. So with that, I know we've had a little bit of technical difficulties, so I want to make sure we have plenty of time to listen to some of the great startups that we'll hear from shortly. So I will stop there and look forward to participating in the rest of the session later on with the panel discussion. So back to you, Philip, I guess. So.
Yeah, perfect. Many thanks, Christopher. And not only the seed is important, but also very important is actually the microbiome is still barely touched area within the whole agri-food value chain. And so I'm happy to introduce and hand over to Alberto, who is not only very familiar in this field, but is also the one you need to ask if you want to know what microbiome is actually around.
Thank you, Philip. Thank you very much to invite me to this webinar. It's a pleasure. Let me share my screen. Maybe we can answer the question with my presentation. Okay, as you mentioned, Biomakers is a US company. We are in West Sacramento, but from 2 years ago, we have also facilities in Europe, in Spain, that we can provide our service from European countries. Well, our ambition as a company is to enable global sustainable farming using a data that is not common. We always say that we can see that others cannot see in the agricultural soils. And we are an IT company. We are developing applications based on microbiome. Well, we are facing some challenges. The global population is growing, and in the last 40 years, we lost 30% of arable soil, including with all the technology that in agriculture work coming in that specific moment. For that, there is a new opportunity to introduce a new variable in all the computing applications for agriculture. We like to introduce the microbiome, because only living things can have health. Microbiome can be a super nice biomarker of soil healthiness. For that, we are decoding microbial interactions in the soil. We are using next generation sequencing and machine learning system to translate the microbes that we are detecting in a soil and use that microbes as a biomarker in a different ways. The first product that we have in the market is BCrop. In BCrop, we are receiving soil samples from the farmer, from the agronomist. We are computing, we are detecting the DNA of that sample in the soil. And we are, with that DNA, identifying bacteria and fungal species that are living in that system. And with that information, we are able to compare at taxonomic level, at functional level, because based on the taxons that are living in the soil, we can understand the genetics of the microbiome. Because don't forget that microbiome is the second genome of the plant and evolve with the plant too. And for the other hand, we are able to compute the ecology. With that, we can translate that information in a different indicators. This is an example of our report, and we provide soil quality metrics, biodiversity metrics at taxonomic functionality level or resistant level, healthiness metric, the disease risk based on the pathogens that are present, but also the ecology in the samples, biocontrol species need to be considered too. Also, some indicators of phytohormone productions or the stress adaptation of the plant that come from the microbiome. And the last one is indicators about nutrition, but not nutrition in the way that we understand the nutrition with a physical-chemical analysis. This is like, based on the microbes, the ability of the microbes to mobilize the nutrients to the plant or to compete and use that nutrients to survive and grow. Well, this is the specific report that we are providing. With this specific report, we can help farmers and agronomists to improve yields, to improve the quality of the crop and on the soil, detect diseases risk, terroir characterization for some crops like vineyards, cacao, or coffee. This is very important for marketing perspective to understand what are the biological variability that can provide specific flavors of characteristics to the end product, and also tracking the effect of products and management systems. Well, this is an example of a report with the index from healthiness that is very low. That means that we are identifying high-level risk of diseases based on pathogen detections. And also we have an online platform that we can draw this biological information on a map and easily compare the disease risks, for example, in this strawberry field. For the other hand, for the nutritional perspective, we are able to characterize all the genes that are involved in different processes of the nutrient cycle. This is an example from the nitrogen. And we can classify the metabolic pathways into pathways that are beneficial for the plant or are in competition with the plant, or intermediate pathways that are involved in the nutrient-nitrogen cycle. In our report, it's very easy to understand. High level means that the microbiome is providing and mobilizing the nitrogen to the plant. Low level is in the opposite way. But also we disclose the different pathways and the results that are providing the overall indicator. Microbiome also is a very nice biomarker of farming practices. This is something that we tested, and as a startup, we like to also provide success case of our technology. This is not magic. When we are computing a conventional, organic, or biodynamic place, for example, in vineyards, but in any kind of crop now, we tested it. And we are developing a network approach. Is the same approach that Facebook or Twitter is using to understand social relationship. We can identify specific patterns. For example, in conventional agriculture, microbiome is very specialized, are producing clusters of activity and functions. Here, H dot is a microbe, and the lines are the relationship between the microbes. Compared with a more organic or biodynamic agriculture, where the specific microbiome is less specialized. This have an impact on yield, but also allow us to establish a rank of the management practice system, because right now in agriculture, we start to talk about regenerative management practice that is in between conventional and organic. It's a kind of practices. Well, how that are improving the soil health? Well, we have a new specific product that can certify how our specific management practice of products are able to impact in the soil microbiome in soil health. The last one that I like to present is Biome. Biome is a specialized product that allow us to analyze the effect of biological products on the field under some specific protocol. Well, we have different examples from Bayer Crop Science, Bioverica, or UC Davis that are using our technology. Here, we are analyzing the effect of the product into the life in the soil that allow to the producers to put functional claims of the product and also understand why the product is effective in some place, under some environment and some microbiome, and in other place, the products are not effective, specialized biologicals. This is another example that we published for one of our clients, that one of a fungicide application, a bacteria that is applied to the potatoes, that the application is a fungicide application, but in some place increase the yield more than 40%. And we use microbiome specialized to predict the effectiveness on that specific product to improve the yield based on microbiome sample. It means that we can take a sample before planting a potato and try to predict the potential impact of the application of a product, and that allow to the farmer to decide if they, for them, is cost effective to apply the product or not. This is available, and also we know the specific microbial properties that are impacting on the yield. Well, for that, with this kind of technology, with supervised machine learning system, microbiome, and other metadata that we can add, we are aiming to test recently the first virtual assistant in order to recommend products and management system based on data, not based on experience. This is a huge effort. For that, we launched a non-profit initiative that is called Fields Forever. In Fields Forever, we are partnership with different companies and universities around the world. There are more than 170 different projects today, and more than 20,000 samples are collecting, specialized in Europe. And to achieve some specific topics, for example, one of them is carbon sequestration, but also soil health, disease risk prediction, or improving yields and getting sustainability. Well, thank you very much for your attention, and if you have any question, please, I'm happy to answer that. Thanks.
Many thanks, Alberto. Please put the questions in the chat, so we will collect them and have a discussion later after the presentations. I would like to hand over now to Martin Kollmar to tell us more about the genetic basis of the plants and how to really get the GEEP side into it.
Thank you, Philip, for this introduction. Thank you for inviting me to present our project here. Let's switch now to the screen. So the project is an Exist-Forschungstransfer funded project at the University of Göttingen, and we recently founded a company that will take over the project and try to bring the product into the market. Let me first introduce our team. There's Dominic with a strong background in IT technology, and then we have Michael with a background in business development and economics, and I have a very strong background as a former group leader at the Max Planck Institute of Biophysical Chemistry in long-time research and annotation of eukaryotic protein families and eukaryotic genomes. This summary slide comprises all the process from during the breeding process. So you start with some germplasm, then you take on a lot of data from phenotype data, omics data, gene function, and combine that to reference genomes. And in the end, you need all that data to do better molecular breeding or gene editing. Molecular breeding means more kind of marker-assisted breeding and these smart breeding processes today. And where we come in is all that in between. So we take all the omics data, gene function data, and map that to genomes and then hand over the data to the breeder. So this is a very nice review. The only thing I don't really agree with is the term reference genomes. The more modern term would be the pangenome. The pangenome concept means that you have a core genome in several varieties or strains, and then around this core genome you have certain strain varieties. And this is not a fake concept. As you can see in this analysis of Brachypodium distachion model grass. So in one genome of this model grass, you have about 25,000 genes. And if you add up to 50 further genomes, you add 30% more genes to that. So the genome annotation process is now to bring all the data onto the genome. So the basic level is the gene locus, where you have on the chromosome some regions where you identify some phenotypic effect. And on top of that, you have this gene structure of every of these genes. And on top of that, further smaller data like alternative splicing, the exons, some SNPs, and structural variations. And on top of all of that, you have the functional annotation that tells you what the structure does, the gene does, the protein does, and all this combines. And let me give you an analog on how complicated it is. So if you take a text and try to translate it with Google Translate, and you take a simple text, then it's similar to a prokaryotic genome annotation from bacteria. You just have a single open reading frame from start to stop, that's it. Eukaryotic genome annotation is more complicated, like in this Roman from Reinhard Jürgel, and you see already in this highlighted text that there are words that Google Translate doesn't know. There's no interpunctuation and all that. So eukaryotic genome annotation is well too complicated and it's not solved yet. I've brought here an example of a myosin gene, and if you look for that in Oryza, then you will find two overlapping genes on even two different strands of that. So there's a lot of noise in the public databases from wrong gene predictions. This is another case from a Hordeum vulgare myosin. And there you see you have many, many transcripts, and all of them are different, have different exons in it. Many exons are missed, different combination of neighboring genes. And all that adds up to the databases to have a lot of wrong transcripts. And these searches were just based on putting in certain gene, in this case, these myosin genes. If you look for just words, for example, if you search for words like myosin genes in this Hordeum vulgare genome, then the results suggest that there are 59 genes in it, but the true number is just 12. So you have a lot of wrong functional annotations in these databases as well. This is just for single cases. If you do add up this to whole genomes and do it yourself in a gene prediction, you can use different software. I've just done this recently for Millerosima acacia. This is a small yeast, and you see already that with 5 different annotation software, you get completely different numbers of genes. In a more bigger genome of a small fungus, Magna Porto, you get also different numbers, and I wanted to present very fresh data, so the two softwares, GeneScan Glimmer, are still running. And there is no development of further gene prediction software since about 2013. If you put all these wrong data into a database, and the database will look like this. Imagine that every of these colored pieces is a different gene. And if it has the same color, then it's about the same gene function, like hexokinase or an ion transporter or something like that. And usually you go into these databases and then you try to pull out the genes of interest with some homology In sequence databases, you will do that with BLAST usually, and BLAST for, let's say, 20 to 30% pairwise sequence identity, and then you get out all the homologous proteins. In this case, it's a picture, so I used Photoshop with a tolerance setting of 32, and that's what you get then. So you get a lot of completely different genes out of that, and as a human being, you will easily recognize that they don't belong together, but the database and the software don't know that. What you really want is a picture like this. So you have all the genes ordered, and you can see which belong together. It's all fine, and you can sort this up. In this picture, every of these columns represent a certain gene with a certain function, like resistance to some insect pest, or resistance to some fungal infection, or a gene for deeper roots or growing of things. And all the lines in that represent the plant species. So if you take just from every column a certain piece, then you will get the genes of a certain species. And from this picture, you will also see which plants don't have certain genes in their genome. And that's a picture that we will give you if you cooperate with us and get a genome annotation with us. I'll present you some fresh data from our new software system. So we did a short analysis of the latest best software to compare our data with. And in this case, you see some genes with some function and some software on the left column, and Scipio version 1.5 was the only software that could reconstruct and predict genes correctly. All others failed in certain cases. So the next comparison is with this Scipio software. And this is a prediction in the fungus Magnaporte oryzae. With Scipio, you get a decent completeness factor. Yes, you can reconstruct 11,600 of, say, about 12,000 genes. And with our new developed software, we don't get as many complete genes yet, so we are still working on that. But very importantly, what you see here is that we get all these alternative fits. That means that are all the genes from the columns in the vertical. So we know already which genes belong together and which have the same function, which have the same gene structure, and which get the same functional annotations in. And very importantly, we get this in much faster time. So we can provide you a genome annotation really in minutes and not in days. And that is not a single case. So we have filled up, or we're starting to fill up our proprietary database with further data by annotating every genome of a eukaryote that is available. These are just some examples for fungi, what we are also annotating the plant repertoire. So that's what we will deliver to the companies who are interested in getting the data combined from phenotype to omics and gene function data onto the PAN genome reference genome, and use that data for setting up better smart breeding and genome editing. Yes, and I will now hand over back to Philip.
Okay, many thanks. So that was a deep dive in the newest annotation project. So many thanks, Martin. I would like to hand over to the other Martin Spitznagel. Martin, can you hear us? You had some issues?
Yes, all
yeah,
good.
perfect.
Yeah, perfect. Thanks, man.
So now the magic camera made disappear. No, you're back. Perfect. Okay, so as I said, questions, please, at the end. We are a little bit late, but we'll just stick a little bit longer on the platform. The platform is recorded, or the webinar is recorded and will stay on the platform. So whoever needs to move away in 5 minutes, then you will be able to get the also till the discussion. So now I'm handing over to Martin, and now we are really touching the ground.
Good, thanks, Philip. Actually, at Bimex, we are accelerating the transition towards a chemical-free wheat management. And we are an industrial biotech company based in that biotech cluster in Munich. Currently a little bit more than 30 people, and yeah, pretty much driven by biotech, by biochemistry, by biology in general. And the reason why we exist, pretty much straightforward. We want to improve sustainability of established industry through biotechnology. That means everything we do has a very clearly defined sustainability component, and we can really measure and show if it's on water, if it's on synthetic chemicals, if it's on CO2. Established industries, we will touch that in a second, and biotechnology, that's our DNA. We wouldn't be able to solve problems in a chemical manner. At the end, we consider ourselves to be experts in biological binding of material. That means our products, in the middle, they either come as a liquid, then they look like a beer. How could it be different? Because where we are based at, or they come as a granulate or as a powder. If you pour that beer over any permeable material, then it will get solidified. And the reason why that technology is also called biocementation, it's because soil bacteria, they are producing a certain mineral, most of the time calcium carbonate. And at the end, this is a natural occurring solidification process that we have industrialized. We have a patent on the technology itself, but we also have multiple patents for applying that technology. First of all, we apply it in the mining industry, and there we sell a dust control and pelletization product worldwide. That shouldn't be the topic today. Secondly, within construction, insulation materials, for example, that's where we have something that's also not the topic of today. And then within infrastructure, for example, unpaved roads, that's something we find quite interesting. That's also not the topic of today. Today's topic is agriculture, of course, and within agriculture, our main focus at the moment is on weed prevention. And within weed prevention, alternative methods on weed prevention. You know all of those approaches, and I just want to make clear that we like those approaches. It's not that we think that there will be one technology ruling everything, and we actually have the big advantage that there's lots of links to other approaches on weed management. So we see that everybody should work together in having, like, at the end, the largest impact possible and as quick as possible. So, but what does that mean? We take our beer in existing equipment, like, for example, that truck. Actually, on the participant list, I just saw someone from Amazon participating. I hope the trademark is okay for using that picture. Or we use our granulates and we put it in the existing equipment on top. But what then happens actually is that we are forming a physical layer within the soil through biocementation that will then prevent weeds of coming through and from competing with the target crop, of course. At the same time, we don't have any negative impact on the exchange of water or gas. And at the end, of course, it is non-selective. That means, like, we don't care about what weeds will come up there. And of course, we can steer it. That also represents a very long-term and at the end, a very natural and sustainable approach to weed management. That's the theory. Let's have a look into practice. That's one example for a field trial on kohlrabi in Germany. Here you can see how we apply it. On the bottom left, you can see the control. So yeah, there's quite some weed pressure, but not that crazy. On the bottom right, you can see the industry standard, and on the top right, you can see our approach. Another example is on potatoes. On the left-hand side, you can see the untreated control, and we just circled the weeds popping up. In the middle, that's the industry standard from one of the big agrochemical companies. Of course, the industry standard works well. On the right-hand side, you can see our approach. In this case, of course, we are adding additional functionalities, and that's, by the way, what we love. It's not about like doing one thing, but having the advantage of using a biological system that can cover many topics. In this case, for example, erosion control, that's automatically integrated with what we do. One more example I brought today. It's quite an ugly slide, but then it's obviously in winemaking, the quality of the pictures is terrible. That's my fault. But on the left-hand side, you can see the standard regime, at least like in the central of Europe. You use 2 times glyphosate and then a mechanical approach on weed management. If you have a look in the middle, then you can see, like, if you don't do anything, then very quickly you will have quite some weeds popping up. And then, of course, we have chosen something very nice and representative. But then if you have a look on the right-hand side, it's pretty much clear. Then there's, of course, it's data-driven. In blue, you can see like glyphosate, 2 applications of glyphosate. On the left-hand side, you can see 3 of our product formulations, which, yeah, don't look too bad compared to the standard. What is our status quo at the moment? We have performed more than 100 field tests, 15 different crops, all over Europe and 1 outside of Europe. Pre and post emergence, of course, there's very often the question, hey, what do you do if you apply pre-emergence? And yeah, most of the trials were also performed with independent service providers and with partners. The value proposition at the end for us, it's we have something that works and that is proven, and that is not only about like doing something similar, but also in some aspects doing it much better. Environmental advantage is that it's a natural process. It's something that happens in nature and has absolutely no negative aspects to the food chain or whatsoever. It is easy to use, so we don't need anything, no equipment, nothing special. And then I already mentioned additional functionalities. Like we have many different aspects that we can achieve with that system. And so our dream is actually like a biological toolbox where many different aspects will be covered. But today we are just talking about weed prevention. Our roadmap currently, we have a product, interestingly, a B2C product that we're selling within home gardening. Then we are approaching applications on railway systems, as you know, like they have quite some pressure there. But then also like having a market entry within permanent specialty crops. We just saw that wine example, or maybe the apple trees on the right here. Then non-permanent specialty crops will follow, and then row crops are on our midterm horizon. If you want to join that journey, then please shoot me an email. We are interested to connect to each and everyone. We are also recruiting, because I saw some participants are also students and other people. We have job openings, and we are always open to partner up. Thanks a lot.
Perfect. Many thanks, Martin. So I actually have no information about how many questions are already in the chat. So I would like to hand over to Chris. Do you have any question? Otherwise, I would take a try.
Sorry, let me unmute. Yeah, I think there's some great presentations, and very appreciative of the breadth across things like weed control are certainly of interest to us, and especially in being able to reduce dependence on chemistry, Martin. So I appreciate the presentation very well. But yeah, no specific questions. I know there's a number of people from our team in KWS that are watching, and so there may be some specifics that we'll all talk about, and may contact some of you individually around that. But I would say great job from my standpoint, and happy to answer any questions that I can regarding KWS as well. So.
Okay, so I have questions, starting with Alberto. So tapping into the microbiome area that has been actually not tackled at all, right, or till a couple of years ago. But what would you say, how important is the interaction between the existing microbiome for the certain field and actually the seed that's being applied or that's being used on this specific field? So would you say that you would have a great impact in actually recommend a certain type of seed or even then also a fertilizer?
Yes, we have 2 projects, at least in Fields Forever, that we are testing different genetics on corn in the north of Italy and also in different regions of Italy. Each location in the world have a different microbiome taxonomy, like animal or plants, if we are comparing a jungle in China with Brazil. There are different taxons at genetic level. But at functional level, they are working in the same way, the same functions that we can able to identify. Well, in that trial in the north of Italy, we correlate that to predict better performance on the crop, the genetic, the seeds plus microbiome are more informative than the microbiome of physical chemical properties alone. Also, another project in UK that we are testing nitrogen mobilization on wheat. They identify some QTLs linked with the genetic of the plant to mobilize or to take more nitrogen that are interacting with the microbiome. And when that happens, there are more nitrogen efficiency in the crop. There are 2 evidence today in our set of data together with our partners and projects, universities and companies, that say that yes, the genetic plus microbiome is very important. But if we look at humans, that's the same with the gut microbiome and the genetics of the persons, for example, the immunity system. For that, there is a lot of correlations because the gut from the plants are the roots, right? And the microbiome in the roots are very important, and the connection with the plant, with the exudates that they are providing to attract the specific microbes that they like to get in touch.
Okay, many thanks. I just move on to Martin Kollmar. So we see that there's a lot of pressure of change on the field. So not only climate change, but also changing actually the ways the soil is being treated, more generative methods. And obviously we come up with the demand on new variants of crops for the maybe different situations. So what would you say is actually your contribution to that? What is the benefit in time savings on a new development, or actually also adding new functionalities within the same time frame of the normal development of a new crop?
I think with our product, so we can add to the time component. So the plant breeders should get much faster, much more new varieties, and it's also then a quality thing. So what I showed in the picture where you have these colored pieces ordered, that means that it's you know a certain phenotype in one plant, let's say wheat, then you can transfer all that knowledge to potato or tomato or any other plant. And at the moment where all these genome annotations are just to 70 or 80% wrong, you don't get that combination. And then you start all the process in every plant again from about 0 and try to get all that functional data collected and all that. And I think another benefit for the companies would be that they don't waste so much of their data that they collect. So they collect a lot of data, images and genome sequence data, and that costs a lot of time and costs a lot of money, and a large part of that is wasted because it cannot be mapped onto the current reference genomes. And with our better data, then you can get more out of the data that you create.
Mm-hmm. Martin Spitzhagen, now to you. First, I need to add also that you're still raising a funding round. So anybody that is interested in that, please give us a notice. And what would you say is the perfect combination, or how would you differentiate between different additional measures of weed control in addition to your method, and how often do you need to apply the BindX technology?
Good question, of course. Actually, one of our big advantages is that the duration is that long. That example on vineyards, we replace 2 glyphosate applications and 1 mechanical one just with 1. So we are covering the entire vegetation period. And that's the thing that we can also steer. If you need a certain effect just probably for a couple of weeks, or you need it for a couple of months, we are capable to do so. And that's also probably the answer to the first part of your question. Very often we have something that works immediately and quite solid, but the long-term effect is not there. That's where we very often jump in. And on the other hand, what is also important that we see, even like if you use the existing chemicals, that there's a big, very, very beneficial combinatoric approach. So if we have a transition period where because it won't be chemicals, yes, no, and there will be some transition. So we can just drastically reduce the amount of chemicals just by adding our biological component. And yeah, that's the reason why we firmly believe it's not like against each other. It should be like a competitive landscape where everybody works together.
Very, very interesting. So I'm eager to see how your technologies, how products are being applied in the current and in the future agri-food value chain. I guess I still have no other questions I'm missing out in the chat.
I have a question for Alberto, maybe. Can you talk a little bit about the impact of spatial variation in the microbiome across the field? For example, how do you characterize one area of the field versus another? Is it just sampling, or how do you, and what is the impact of that, or have you seen either from the markers that you might identify across a hectare of a field?
Perfect. Yes, microbiome is very sensitive and is collecting very good environmental conditions, specialized. Well, there is always variability in microbiome. To collect a sample, we need to get a representative sample of an area. For that, we are taking core samplings to the same tube to analyze the sample. That's essential. And the variability, for example, that we can identify during the season is, because there are variability in the microbiome, is less than the variability that we can find from one sample to another kilometer far. And depending of the location of the sample, they can change more or less. They're always changing. But yeah, the geographic location is one of the drivers, because we know perfectly what are the specific drivers in microbiome. For example, pH is the main driver. Second is the organic matter, content of water, redox, and also the plant variety. That's something that was a surprise for us and push us to develop crop-specific database of microbiome, because each plant is attracting a specific group of taxons because they have different genetics, different chemical exudates, and different way to exudate that chemicals. For that, we develop all our database independently per crop. But that's for the taxonomy. Again, taxons, yes, are different. But functions, there is only a pool of functions that we can play, that are all the genetics that are describing for the genomes of the microbes that we know, okay? And that the amount of functions is limited right now, compared with the amount of microbes that you can identify. For example, we know in the database that are public, there are 350,000 different bacteria with names, species level. In our database, we have 5.5 million of reference of DNA chains. Some of them, we can approximate the reference to the genus level or the species level, but sometimes are unknown. That's the diversity of the life that we can find in the soils.
Thank you. Yeah, and I can imagine that with crop rotation, if you're talking about specificity, then these change year over year. There's some variation and temporal variation year over year.
For example, we tested the rotation of corn and soybean in Brazil. And one of our first hypotheses, okay, maybe the microbiome don't change a lot between corn and soybean, but not. If we compare a pool of samples from different places in Brazil, corn and soybean, microbiome looks totally different in a principal component analysis, in a beta diversity analysis. That allows us to say, okay, no, the plant influence is huge, in this case, in the rotations. Also, if you are improving the diversity of plants that you are putting, for example, adding cover crops or other things, are increasing diversity because there are other plants that are attracting other microbes.
And even weeds, I would imagine, would impact that as well.
Yes.
Thank you.
So, very interesting discussion. I could stick to it, but I guess we need to close. Slowly, we need to close this webinar since we are a little bit over time. Many thanks to you all for presenting your companies, your technologies, your products. I experienced a very good time. That was a showcase on what for technologies we love, how we want to interact with the startups, with the corporates, always on eyesight. And I hope that we meet all once in Hanover, at least during the Agritechnica. And whoever has questions, please reach out to us. And for all the participating startups, we have an Ask Me Anything session for the application process of the RootCamp. That will be on the 13th of August. Please note that the application closed on the 17th of August. And I guess it became clear that we love to see knowledge being applied in the fields. And if there's some deep tech around, we also don't mind that. So many thanks again for participating, and we hope that the discussion continues. So
thank you for having us.
Thanks a lot. Bye.
Thank you very much.
Thanks a lot.
Thank you.